Hi everyone, welcome back. When bacteria reproduce, they copy their genome exactly, or at least that's the intention. Because these biological processes are certainly capable of error. 99.999% of the time, this effort to copy perfectly is successful. But it's that 0.001% of the time that a mutation can occur. These mutations are any change to the genome that could potentially change, if it occurs in a coding region, what protein it encodes for, or what that protein looks like when it's produced. These mutations can be positive, they can be negative, or more often than not, it doesn't have any impact on the organism at all. Now, this is dependent on a variety of factors, but one of the major ones is the environment. Traits that could be good in one environment could also be bad in a different environment. It's about whether or not that trait is useful to the organism in that environment. Even a mutation like antibiotic resistance can be useful to a bacterial cell if that cell is never exposed to that antibiotic. The environment is the decider about whether a trait is useful or helpful or harmful. But in the right environment, specifically one that contains antibiotics, that trait of resistance is really beneficial. These mutations emerge randomly and rarely. Importantly, the cells do not choose to mutate. They don't mutate in response to anything. Mutations occur completely randomly. But if the environment is right and that mutation ends up being helpful for that organism, that individual with the mutation can survive and reproduce when others cannot. Now, antibiotics, when they are in the cell's environment, they exert an evolutionary pressure on the bacteria who live in that environment, who experience those antibiotics. This is a method of selection in the population. Vulnerable cells tend to die when exposed to these antibiotics or fail to reproduce. Resistant individuals tend to live longer and reproduce more. Slowly, over time, these populations are shaped by the survivors, the ones that live and the ones that manage to reproduce. So in an environment with lots of antibiotics, we are going to see a shift in a population of bacteria towards being more resistant, less vulnerable. This is a natural force, something we call natural selection, and it's really hard to fight against. It's something that occurs in all living things. All populations experience natural selection in one form or another. So this is a major challenge for modern medicine when we want to use a drug that exerts a significant selective pressure on these bacteria that cause disease. For example, take that beta-lactam ring and the drugs that have that, these penicillin-type drugs. This ring is the effective part of that drug. And some bacteria have evolved resistance to penicillin and these beta-lactam-type drugs. They've evolved whole new enzymes in some cases, like beta-lactamase. This is an enzyme that will actually attack and break apart that ring, upsetting its key function as an antibiotic. That is one mechanism of resistance. There are many more that have evolved. And this is important to recognize too, because there is not one right answer to every evolutionary problem. A cell could deal with an antibiotic as a problem in a variety of ways. A resistant strain might have evolved the ability to deactivate those drugs using special enzymes like beta-lactamase, but it might have also evolved a strategy to alter its own enzymes so that those drugs can't bind to it anymore. It might be better equipped to remove that drug from the cell once it gets exposed. Or perhaps it has a strategy to prevent uptaking that drug in the first place. There's a diversity of strategies that are possible, and evolution can come up with many different solutions to the same problem. This is really important because it means that antibiotic-resistant strains are not all the same. They don't all have the same mechanisms to fight a specific antibiotic. Oftentimes, if they've evolved it independently, their solutions to that problem are completely different. Many different kinds of mutations can result in that antibiotic resistance, and some can confer greater resistance than others. And you may even have situations where bacterial cells have evolved or collected multiple mutations that are solutions to the same problem, and that can amplify that resistance quality. Now, mutation and selection together can shape these bacterial populations really quickly. Evolution is something that occurs on the timescale of generations, but bacterial generations are really short. It still takes many generations to see meaningful, significant change in the population. But when a generation takes only hours or days, only a short amount of time is needed for many of these generations to pass. However, resistance is not the same as immunity to a drug. Resistant strains can still be vulnerable to higher concentrations of a drug or different methods of drug application. And we can also modify these different treatment strategies to deal with drug resistance. We have to modify in order to deal with already resistant strains, and we need to make sure that we can prevent new resistant strains from emerging or make it harder for them to evolve in the first place. So here are some different solutions to that. One strategy is to apply multiple drugs together to handle particularly tricky situations. So when we take multiple antibiotics and use them together, we could experience an additive drug action where the different drugs sum together for an effect. It's kind of like if you imagine this as an addition equation, like if you had 3 plus 5 equaling 8, where 3 and 5 is the power of these two different antibiotics that you sum together. And together, they have an impact of 8, a stronger impact, but it's really just summative. Synergistic drug action is considerably better. This is when you have 2 or more drugs come together to have a multiplicative effect. So you're no longer adding, you're multiplying. 3 times 5 is 15, a much greater result than summing these 2 drugs together. Some drugs work together additively, and some drugs work together synergistically. These drug action strategies can let us reduce the dosage of each drug to cause a significantly greater impact. It also can help us avoid the harmful side effects that can sometimes arise, or the loss of selective toxicity that can occur with high doses, super high concentrations of these drugs. I also want to make sure that we're familiar with some of these other terms that are pretty important. We have minimum inhibitory concentration, or the MIC. This is the concentration of the drug that is needed to inhibit bacterial growth. Minimum bactericidal concentration, or MBC, is the concentration of a drug needed to kill bacterial cells. These are pretty important concepts. And so if we apply these back to our idea of additive or synergistic drug interactions, particularly with synergistic drug interactions, we can use multiple drugs together to reduce the relative minimum inhibitory concentration or bacterial concentration for these drugs when they're used in tandem.